So this video is entitled Basic Experimental Design. So we're going to move past the pre-planning process into the actual design of the experiment. So again, I'm going to reiterate why we're even designing the experiment is so the student researchers can make valid and objective conclusions. So we're going to again ask them to ask the right questions, which we did in the last video. And now we're going to make sure they collect the right data. So phase one is the pre-planning process, and that is done before designing your experiment. So they've already developed a testable research question, they've already identified their independent and dependent variables, and they've already identified the other variables that might impact that dependent variable. So they have already identified all of those key aspects that'll help them design their experiment. So in the actual experimental design, they're going to understand how basic design principles work. They're going to be able to identify key aspects in the planning process that transfer and dictate the design. And they're going to ultimately sketch out their experimental design by the end of these activities. So they're going to translate the things that they identified in the pre-planning process into the actual design, and they'll learn how to control for those other factors. They'll learn how all of those pieces start to come together in a design of experiments. I wanted to provide a quote by Doug Montgomery, a very famous design of experiments expert. All experiments are designed experiments. Some of them are designed well, and some of them are designed really badly. The badly designed ones often tell you nothing. So we want to avoid students falling into that trap. We want to avoid getting them to a situation where they can't make those conclusions that they want to make about their research question. So it is important to design their experiment well. So we've moved beyond the factor identification and the hypothesis identification to experimental design. So we're in the second aspect of our from planning to conclusion process. The key experimental design aspects that we're going to talk about are randomization, replication, and comparison. So the first one is randomization. We randomize to prevent selection bias. So selection bias means that one group is different than the other group by some way I've selected the two groups. So if, let's say I'm testing an additive versus a non-additive. I need to make sure that my experimental units going into each group, either they're getting the experiment or they're getting no experiment, they're the control group. I need to make sure they're equal in all other aspects. They need to be the same in all other aspects, and how we do that, typically with human trials, things like that, with people, with animals, with things that we have natural variation, we want to randomize them to two groups. So we will randomize your experimental units into the control group or the experimental group. They'll either get the treatment or they won't. But the key here is that if you randomize into one group or the other, then we mitigate all of the other bias that could come from the factors that we can't control. So typically these are for human factors, animal factors, things like that, that have natural variation. Variations in weights, variations in heights, variations in other things that may impact your dependent variable that you just don't know about. That's why we randomize into two groups. There are situations where you don't actually need to randomize. Sometimes in an experiment, we already know that all of our things are the same. For example, we order cells, and the cells are the same no matter if we put them in the control group or we put them in the experimental group. So regardless, they're going to be the same. So we don't need to randomize into those two different groups. So really the randomization comes in when you're talking about people, you're talking about animals, things that other factors can impact. So the second experimental design basic that I want to emphasize is replication. So what we're calling replicates, the number of experimental units that you have. So more replicates provide better information. You can't just do the experiment on one person or you can, but it just doesn't provide as much information. So it's better to include more people, more cells, more whatever you're using as your observational unit. You want to include more of them. You want to replicate your experiment to see if you get the same results, or maybe you're getting different results. So obviously, the higher the number, the better. But this is often constricted by budget and time constraints. So even though there is a statistical way using a power analysis to actually figure out how many people or samples you'll actually need, that's usually out of the scope of an iCure. We'll just usually stick to time and budget constraints, and you're just going to get as many as you possibly can within those constraints. Finally, we're going to compare. So the last aspect of experimental design would be to compare the two groups. We want to give them different treatments, and then we want to compare. So we want to randomize, replicate, and compare. So in the activities, we provide students with some simple experimental designs that likely their experiment is going to take. So hopefully they'll be able to identify the design that's best suited for their variables. As a reminder, students already have identified their variables of interest and their response variable. So they have already identified all the key aspects to this experimental design. So you start out with the same units, then you randomize into two different groups. One's maybe manipulated, one's not manipulated. And that's manipulated by your factor of interest. Okay, so that grouping in this center is your factor of interest. Then you conduct your experiment, and as a reminder, you've already identified these variables. As a reminder, you're going to need to measure your dependent variable, and you're going to measure the other variables that you decided that you've identified in the beginning in the planning process that could potentially affect your dependent variable. And then ultimately, you're going to compare. So another design that's fairly typical in an iCure is a pre-post design, where you start out with all of the same units, you apply the treatment, and then you compare them. You collect the information and then you compare them before to after. So for example, we give the students an example in an activity. So we ask them to identify all these different factors and insert them into their experimental design. But part of that idea generation is to show them a simple experimental design so they can start to see how their experiment will compare or contrast to a simple experimental design. So, for example, this research question is, does a new medication lower the low-density lipoproteins, or the LDL levels of cholesterol, more than the current medication, controlling for diet and weight? So this is a nice example where they've already identified the dependent variable, the cholesterol level. That's that objective measure of whether or not the drug worked or not. Our factor of interest would be the drug, and we're going to split that factor of interest into a control group, the old drug versus the new drug. And then the last statement states controlling for diet and weight. Those are those other factors that the student identified as potentially affecting your dependent variable that they're going to measure. So they start out with their experimental units. They randomize into two different groups. One group gets the old drug, one group gets the new drug. You'll notice that I added the sample size with the number of students in each group, so the students start to think about how many replicates they're going to need for that part of the experiment. Then they're going to conduct their experiment. So then they're reminded in conducting the experiment to collect the response variable and to collect all of those other variables of interest, i.e. here, the diet and weight variables. And then ultimately, they're going to compare the cholesterol levels between the old drug and the new drug, and that comparison will help lead to their conclusions.